Hi everyone, this is Xiao from UPenn. Thanks for the invitation. My lab is focusing on therapeutic editing for the correction of pathogenic mutations responsible for metabolic disorders, and I'm excited to share our work with you today. First, I will start with PKU. PKU is an autosomal recessive disorder caused by mutation in the gene encoding PAH. PAH is responsible for breaking down the amino acid phenylalanine, and loss of function of this gene results in the accumulation of phenylalanine into neurotoxic levels. It's estimated that correcting just 10% of the allele would be enough for therapeutic effect. The key to treating PKU patients is reducing blood Phe levels. The mainstay treatment is a low-Phe diet, which is difficult to adhere to, followed by an oral medication that doesn't work for many of the pathogenic variants, and an injectable enzyme that's taken daily and carries a substantial risk of anaphylaxis, causing it to have a black box warning from the FDA. All of these are chronic interventions that one has to take for a lifetime. So what if we could develop one-and-done genome editing therapies that could directly correct the pathogenic mutations? More than 1,000 PKU variants have been reported, and of them, the most frequent variant worldwide is R408W variant. Using data from patients with PKU treated at the Children's Hospital of Philadelphia, we found that the majority patients had at least one single Phe measurement above the recommended safety zone indicated by the dotted line here. About 30% of the patients had a lifetime average Phe levels above the recommended maximum Phe levels. This emphasizes the significant unmet medical needs for PKU patients to better control their Phe levels. Then, what genome editing tools do we have? I believe the audience here are familiar with Cas9 nuclease editing and base editing. Very briefly, Cas9 protein can bind a specific DNA sequence and introduce a double-strand break. The default repair pathway in cells is non-homologous end joining, in which the free ends from the double-strand break are rejoined. This is an error-prone process that results in the insertion or deletion, collectively termed as indels. If the double-strand break is directed to the coding region of a gene, it would most likely to disrupt the gene. On the other hand, base editing strategy offer the advantages of precision editing. With base editing, only one strand is nicked. We could introduce C2T, A2G, and C2G changes depending on the selection of base editor. Prime editing is a search and replace technology. The prime editor protein is a fusion of a nickase Cas9 and a reverse transcriptase. The pegRNA contains a programmable RNA template and a primer binding site. The 3' RNA extension of the pegRNA can introduce process changes to DNA, such as base substitutions, small insertions, and deletions. So here are the most frequent pathogenic mutations identified in PKU patients. If you look at them closely, each of them could potentially be corrected by adenine-based editing, A2G edits, either on the sense strand or the antisense strand. First, I'm going to show you our progress in correcting the two variants highlighted in red here. The first one is R408W, which is the most frequent pathogenic mutations in US. The second one, I will refer to as P281L, is the top 6 in US. to show that any combination of base editor and guide RNA actually works well to correct the pathogenic mutations in human hepatocytes. We would need hepatocytes that harbor the mutation, which of course isn't really feasible if one's looking for primary human hepatocytes with a rare patient-specific mutation. So our workaround here was to use prime editing to knock in the mutation into both pH alleles in human H7 hepatoma cells. As you can see here, we were able to precisely knock in each of the variant into H7 cells and obtain homozygous mutant cell line. Now that we have the P281L homozygous cells, we can easily screen editing activity with different editing base editors in combination with candidate guide RNAs. The green portions of bars here in the graph indicate outcomes with the desired on-target editing, the correction of the P281L mutation, and the red portions of the bars here indicates the undesired bystander editing. As you can see, I highlighted the best results here, which is with ABE 8.8 and guide RNA 1. We used the same strategy and found that spry ABE 8.8 along with a guide RNA putting the variant at position 5 demonstrated the highest on-target correction with minimum bystander editing. Now recall that to achieve a therapeutic effect in patients, we think we only need to achieve about 10% correction, so this data are encouraging. We formulated LNPs encapsulating the editor messenger RNA and the guide RNA, and then treated the homozygous mutant cells. As you can see here, we achieved high level of on-target editing with minimum bystanders. Will it work in vivo? The only way to test that is to use an animal model that carries the mutation. We were able to knock in each of the variant embedded within a short humanized segment of the pH locus. We can immediately tell that our humanized mutant mice have PKU because they have hypopigmentation, just like we see in patients. Hopefully, you can appreciate the lighter coat color compared to the control littermates on the right. Our humanized mutant mice have elevated blood phenylalanine levels, just as we see in PKU patients. As you can see with the orange and green dots in the 1,000 to 2,000 micromole range, while the control mice, the purple dots at the bottom, all have normal Phe levels at around 100. Then we administered the LNPs encapsulating the base editor messenger RNA and the guide RNA to some of the PKU mice, the ones indicated in orange here, with the green ones being untreated PKU mice. We found that within 48 hours, the phenylalanine levels had been fully normalized. These are the results from an ongoing long-term study where the normalization of Phe levels has been stable for more than a year so far. We fully expect that the effect will be durable and will represent a definitive treatment or even a cure for PKU in patients bearing at least one copy of this pathogenic variant. I want to briefly shift gear and talk about prime editing for correction the R408W variant, which we pursued in parallel with the base editing strategy as an alternative editing strategy. Using the same homozygous R408W cell line, we screened various PEG RNAs and nickel guide RNA combinations for corrective editing of the R408W variant. And then we treated the R408W homozygous PKU mice with a dual AAV vector system expressing the most efficient prime editing configurations we screened from the cells. AAV takes longer to work than LNPs, but here we see that by 3 weeks post-treatment, there's normalization of Phe levels in the treated mice with up to 52% whole liver editing. So we have 2 viable therapeutic strategies for the R408W variant. Then how about off-target editing? So here we use a strategy named as oligonucleotide enrichment and sequencing 1-seq. So I don't want to get into too much technical details here. Very briefly, 1-seq uses a synthetic library of oligos selected based on the sequence similarity to the on-target guide RNA, and the oligo libraries will be treated with the genome editor, and the cleaved sites will be identified by NGS. To assess the off-target profile of our P281L treatment, we performed 1-seq and then checked the top 100 candidates nominated by 1-seq in the homozygous cells treated with the editor and the guide RNA. As you can see, there are 4 low off-target sites identified by amplicon sequencing. Although none of those off-target sites appear to be biological significance, but ideally, we would like to completely eliminate all of the off-target editing. It has been reported that using RNA-DNA hybrid guide RNAs with Cas9 could maintain the on-target editing while reduce off-target editing. So there's a key reference at the bottom left. We applied this approach to the PH1 guide RNA, and so far we tested more than 40 different RNA-DNA hybrids. As you can see, the 20 nucleotide spacer sequence of PH1, where the orange circles represents RNA bases, our hybrid 27, the DNA nucleotide at the position indicated by the purple squares, was quite effective at reducing off-target editing. Hybrid 27 not only maintain on-target editing, as you can see on the left, the purple versus the orange, but it also reduces editing at each of the 4 off-target sites to the background levels, the purple versus the green. So we've effectively eliminated the off-target editing. We tested LNPs with AB8.8 messenger RNA and the hybrid 27 guide RNA in PKU mice, and we found it works just as well to reduce blood Phe levels as the standard guide RNA. The hybrid guide RNA actually shows better whole liver editing with a mean of 51% compared to 39% with the standard guide RNA. This also, as a bonus, appears to be reduced by standard editing, which you can barely see there in red. We performed a dose-response study of the AB8.8 hybrid guide RNAs using homozygous mutant mice. We found that LNP's doses down to 0.25 mg/kg results in complete to near normalization of blood Phe levels. Across the dose range, there was minimum bystander editing compared to the desired corrective editing. Then how about other variants? So how do we rapidly develop an acting treatment for any PKU patients in need? So first, we would need hepatocytes for each variant to screen base and prime editing solutions for the corrective editing. But it's not practical to do one variant at a time. Therefore, we are actively establishing a pipeline to assemble a series of multiple variants, including P281L and R408W as our standards. These variants will be inserted as a unit into a lentiviral vector, and then will transduce HH7 cells and screen editor and guide RNA there. Using this platform, we rapidly screen base editing solution for 2 more pH variants. We used the corrective editing of the P281L and R408W as standards, so any editor and guide RNA combination that can achieve the editing levels of the R408W variant would be predicted to work well in vivo. So as you can see here, I highlighted the best results here for the variant 3, which is better than the one of the standards P281L. Also for variant 4, we did the same screening and identified an editor and guide RNA combination works better than P281L. So right now, we are also working on a group of devastating, ultra-rare inborn errors of metabolic centered in the liver, the urocycle disorders, and organic asthenias. We began to track each patient diagnosed with one of these diseases by newborn screening at the Children's Hospital of Philadelphia. Here are the diseases and genetic variants of the first 4 patients we identified. For each of these patients, a liver transplant will be therapeutic, but a one-time liver-directed base or prime editing therapy would be better. We would like to apply our PKU workflow to these patients, but we realized that we would need to escalate and scale up the whole discovering process. We need mouse models to assess in vivo editing, but again, it's not practical to do one mouse model at a time. So we've also began rapid generation of mice for testing in vivo corrective editing by integrating the genomic sequences of a set of variants as a block into the ROSA26 safe harbor locus. We've already made the mouse for the variants from the first 4 patients I just mentioned earlier. With this platform, we can develop gene editing therapies in a matter of months rather than years. Utilizing the lentiviral pipeline previously described, we screened various pegRNAs and guide RNA combinations to assess the corrective editing of the alpha variant. We then packaged the most efficient prime editing configurations into a dual AAV system and administered into the mouse model where multiple variants were inserted into the ROSA26 locus. In a pallet low-dose experiment, we achieved about 5% on-target correction. And with that, I would like to thank everyone in the lab, especially Dominique, Madeline, Aiden, who take leads of the projects I just talked about today, and our wonderful collaborators at the CHOP. And we're happy to collaborate with any physicians who have patients with these diseases. And also, I would like to thank the funding resources. So this work is funded by NIH Somatic Cell Genome Editing Consortium. And thank you.